Hello, everyone. It's really exciting to have you here today for our inaugural Units of Life seminar series. I am very much looking forward to launching this series, even in this virtual format, even though we had hoped, of course, to do this in person. The purpose of our Units of Life seminar series is really to respond to a need for, having a forum for bringing new voices from outside our community into our community, scientists who are interested in mechanistic biology, emphasis on reductionist approaches and cellular mechanisms, and trying to build community around these ideas in a very broad sort of setting. Of course, the efforts of Units of Life seminar series is highly complementary to a whole host of other seminar series at the Broad, including efforts in the metabolism program, CCNE, immunology, the infectious disease program, ChemBio, just to name a few. So we hope that our efforts here in our speakers series will be complementary to that. We would like to explicitly focus on scientists who are from outside our community in every way that we can. And of course, when we were hoping to have this seminar series happen in person, we were hoping that people would come from around the country and even possibly internationally, so that we can hear and get to interact with tremendous scientists from all around the world. But of course, we have amazing scientists right here in our backyard, and today's speaker is exactly that, a young, amazing scientist, Lindsay Case from MIT, who I'm going to introduce in a minute. But it's just so exciting to be able to have, as part of the seminar series, a focus not only on scientists who are not traditionally in our community, but also people who are starting out in their careers. Diversity is an important consideration, so we can bring in diverse voices from everywhere. And despite the pandemic and the Delta variant sort of changing our plans a little bit, we do have an amazing lineup of speakers. Some of them are known to our community because we had to draw from people who are closer by. That was when we initially thought the seminar series would be in person. But now we're back to the virtual format, and I'm just delighted that we have a whole host of speakers coming up later this year who are incredibly inspiring in many different ways. For example, Lindsay Case hails from UT Southwestern, where she has done incredible work with Michael Rosen. And later on in our seminar series, we'll hear from a very inspiring scientist from UT Southwestern, Helen Hobbs. And we have a whole lineup of additional incredible cell biologists from the Rockefeller and Yale. So it really promises to be an exciting fall and spring, and looking forward to everyone's participation and questions and suggestions for how we might make Units of Life more interesting to our community. So without further ado, I would like to introduce Lindsay now. She's an assistant professor at MIT. It's just remarkable that she really has started her lab coming from UT Southwestern in the middle of all of this that we're all dealing with. And along the way, she has managed to be named Damon Runyon Breakthrough Scientist, huge recognition. And I was also particularly tickled by the fact that she had received the Brand Goldstein Prize at UT Southwestern, which is actually an honor reserved for the most prominent and most successful postdoctoral scientists at that institution. So it's really a tremendous honor. So as I mentioned, Lindsay trained with Michael Rosen, and she's done very interesting work in phase separation for signal transduction. I think we're going to hear some of her interests in that area today. And I think in her lab, she's hoping to continue on this work, building signaling pathways in a reductionist approach from purified proteins that are coming together, so that we can really learn the elements that mediate important signaling pathways, such as mechanotransduction, and then also liquid-liquid phase separation in signal transduction, what we can learn about that, especially at the plasma membrane. As someone who, like myself, I have worked on membrane proteins for my entire career so far, it's just really exciting to learn from Lindsay about how things are organized at the plasma membrane and how signal transduction can be seen in its most fundamental elements. So Lindsay, thank you very much for agreeing to be our inaugural speaker, and we very much look forward to your presentation today.
Thank you so much, Anna, for that lovely introduction. It's a huge pleasure to be here today, even if in this virtual format. Let me share my slides. Yes, so so I'm new to the Boston area. I, as was mentioned, moved in the middle of the pandemic to start my lab at MIT in January of 2021. And I'm really excited to have this opportunity to sort of share my work with some of my neighbors outside of MIT today at this seminar series. And my research is really focused on understanding the spatial organization of signal transduction. So how are molecules organized at the plasma membrane to regulate their signaling and biochemical activity? And during my postdoc with Mike Rosen, I was really focused on understanding the particular role of protein-driven phase separation in regulating organization at the membrane. And so I'll be telling you about that work today. In my talk today. So my research is broadly interested in transmembrane signaling, which is really a fundamental cellular process that is essential for life. All organisms need to be able to sense information in their environment and respond appropriately to that information. So I'm showing here 3 examples of complex organismal biology that occur in response to fluctuating environmental conditions. In response to starvation, dictyostelium will switch from a single cell stage of the life cycle to a multicellular stage of the life cycle. During development, axonal guidance is mediated by sensing and responding to a combination of attractive and repulsive cues in the environment. And many plants are able to sense the source of light in their environment and grow in direction of that light to aid in photosynthesis. So in all of these examples, there are cells in these organisms that are sensing some change in the environment, and they are initiating a cellular response to adapt to these changes. And multicellular organisms also further rely on signaling for intercellular communication, both through the secretion of signaling molecules like hormones or growth factors, and through cell-cell contact based communication as well. And this process of sensing and responding to cues in the environment is primarily mediated by transmembrane proteins, receptors that are embedded in the plasma membrane. So these are proteins that have some extracellular domain that sense the signal and an intracellular domain that initiate the intracellular response to the signal. So this cartoon from molecular biology of the cell is sort of a simple textbook understanding of signal transduction. A ligand binds to the receptor, and perhaps this induces some conformational change in the receptor, leading to recruitment and activation of an intracellular signaling protein. This will then initiate some signaling cascade that will ultimately change the activity of an effector protein to directly alter something like metabolism or gene expression, or the cytoskeleton to change the cell shape. So this cartoon is accurate in that each of the steps, sorry, let me turn my phone off. Each of the steps depicted is important for signal transduction, but this cartoon also doesn't tell the whole story. So one misleading aspect of this image that has always frustrated me is that we're looking at a single receptor in an otherwise empty plasma membrane. Really reflect our understanding of how molecules are distributed on the plasma membrane. And this is actually important because we know that receptor organization is often a really important point of regulation for many signaling pathways. So the plasma membrane is not this like empty lipid bilayer with a single protein in it, but rather it is a really complex, heterogeneous, and dynamic signaling platform. There can be hundreds or even thousands of different classes of receptors expressed on the surface of mammalian cells. These receptors are very densely packed, and they're also often rapidly diffusing within the fluid lipid bilayer. And this is the sort of spatial or physical context in which signal transduction occurs. So how do cells ensure in this really dynamic and heterogeneous environment that when a ligand is present, the correct receptor and the correct downstream pathway is becoming activated? Of course, cells have evolved many mechanisms to regulate signaling, but we know that one strategy cells use is to actually organize receptors on the plasma membrane. So we know that these receptors are not randomly distributed throughout the membrane, but are often organized and colocalize with a specific subset of molecules. So signaling is often regulated by higher order organization of these receptors. Receptors will often cluster together, perhaps tens or even hundreds of receptor molecules can cluster. They sometimes will prefer a certain type of lipid. They can recruit specific cytosolic adaptor proteins and signaling molecules to colocalize with them to form fairly large domains on the plasma membrane that have a really specific molecular composition. So we know that signaling can often be regulated by receptor clustering and higher order supramolecular complex assembly. So in the case shown here, this is the example of receptor tyrosine kinases. Ligand binding induces dimerization, and the dimer is actually the active form of the receptor. But in this case, even the formation of dimers or small oligomers is still not sufficient to induce robust activation of downstream signaling. Rather, in many pathways, higher order assembly is required, where tens or hundreds of receptors cluster together, recruit many different adaptor proteins from the cytoplasm to form a domain on the membrane that can be hundreds of nanometers in diameter. And it's the formation of these supramolecular complexes that actually can robustly and sensitively activate downstream signaling. So for receptors that are regulated through this type of higher order organization, a really critical point of regulation is this transition from perhaps a small dimer to a more higher ordered supramolecular cluster. So an important question to understand is, how do receptors form these types of micron scale clusters? And this is actually a really challenging biophysical problem for the cell, because these clusters are orders of magnitude larger than individual proteins. So just to give you a more intuitive understanding of this, if this circle represents a supramolecular cluster that's 500 nanometers in diameter, which is a pretty common size cluster to observe in many different cell types, a single individual protein would be the size of this very small circle. So somehow cells are able to use these relatively small molecular building blocks to rapidly and reversibly assemble large supramolecular signaling clusters. And so I became very interested in understanding what are the mechanisms that regulate this assembly, and how do cells control this type of organization at the plasma membrane. And so during my postdoc, I became quite interested in the phenomena of phase separation, because phase separation is one mechanism that can very rapidly and reversibly drive this level of higher order organization. So phase separation has really emerged onto the scene over the last decade as a really common and potentially quite important mechanism for organizing molecules in cells. So phase separation is simply the phenomena that occurs when it becomes energetically favorable for molecules to demix. If you dissolve a molecule in solution, under most conditions, it will be a single, it will exist as a single uniform phase. But under certain conditions, at the right concentration, pH, temperature, and salt, it can become more favorable for the molecule to demix and form two discrete phases: a dilute phase where the molecule is depleted, and a condensed or concentrated phase where the molecule is highly concentrated. And if both the dilute phase and the condensed phase have liquid-like material properties, we refer to this as liquid-liquid phase separation. And liquid-liquid phase separation results in the formation of these liquid-like droplets in solution, much like when you add oil and vinegar together, you can observe oil droplets forming in vinegar. So this is a phenomenon that is quite common throughout the natural world. It's not unique to cells or biology, but we do see it quite often in biology. And as I mentioned, there are many factors that influence whether or not a solution will demix and undergo phase separation, things like temperature, pH, among others. But we also know that one factor that can influence phase separation is oligomerization. So oligomerization, so the conversion of molecules from being monomeric to oligomeric, can promote phase separation because it reduces the solubility of the molecule. So if you have a red molecule dissolved in solution, each monomer can sort of freely tumble and diffuse in all directions. But if you start to cross-link monomers together, individual monomers become restricted in their mobility. They're not able to diffuse as freely. And as you continue to cross-link these molecules together, at some point, you can actually reach a threshold where it becomes more favorable for the entire oligomeric network to condense out of solution and undergo phase separation. So oligomerization, we know from polymer chemistry, is one factor that can promote phase separation. And we think this is important in biology because this is a really common change that can happen in molecules. So over the last decade, scientists have found that there are many biological molecules that can phase separate at physiological conditions. So here, these are examples of molecules that have been extracted from cells, so purified molecules that are combined in a test tube at physiological concentrations with physiological buffer. And what you often observe is that when you combine these certain molecules together, they spontaneously form liquid droplets in solution through liquid-liquid phase separation. And we know that there are a lot of different types of molecules that can undergo phase separation, including proteins and nucleic acids. And so on the surface, a protein is very different chemically from a nucleic acid, so it may seem like these molecules are chemically quite different. But when we look further, we are beginning to understand that often molecules that undergo phase separation share a certain feature. So they have this ability to form multivalent interactions. They have multivalent binding domains or weak multivalent binding motifs connected by very flexible linkers. And this type of organization enables the formation of higher order oligomers. And it's this oligomerization that often can induce or promote phase separation in these biological contexts. So often multivalent interactions of biological molecules, including proteins, can promote phase separation. So the prevalence of this phase separation has led to the hypothesis that phase separation is an important organizing principle in eukaryotic cells. And we're beginning to see more and more compartments, membraneless compartments that form through phase separation. So compartments like the nucleolus and the nucleus, P-bodies and stress granules in the cytoplasm. There's growing evidence that many of these membraneless organelles actually form through the phase separation of their constituent components. And compartments formed through phase separation share certain features. They are often discrete micronscale compartments that lack encapsulating membranes. Their formation is dependent on specific multivalent molecules that undergo multivalent molecular interactions. They can form over a really wide range of molecular stoichiometries in contrast to a molecular machine like the proteasome. And they often exhibit liquid-like material properties because they form through liquid-liquid phase separation. So my research has really been focused on understanding whether this phenomena of phase separation might actually impact the organization of molecules at the plasma membrane. So while these phase separation can form three-dimensional droplet-like compartments in the nucleus and the cytoplasm, the same physics can also drive receptor clustering at the plasma membrane. And so I've been very interested in understanding if there are any functional consequences of phase separation for downstream signaling. So does protein phase separation regulate receptor clustering and downstream signaling? So today I'll be telling you about two different projects that I have completed mostly during my postdoc at UT Southwestern, some of which I'm continuing in my own lab to follow up some studies on. So for the first half of this presentation, I'll be telling you about published work investigating the role of phase separation in regulating actin polymerization downstream of the cell-cell adhesion receptor nephrin. And towards the end, I'll tell you about my more recent work investigating how phase separation contributes to clustering and supramolecular assembly of the cell matrix adhesion receptor integrins. So nephrin is a molecule that some here may be familiar with. It is a cell-cell adhesion protein that's really critical in the kidney podocyte. Nephrin has a very large extracellular domain that interacts in a homotypic fashion to drive cell-cell adhesion. And the extracellular domain is really critical for the formation of the slit diaphragm. So it is the extracellular domain of nephrin that really contributes to creating a molecular filter that can filter blood in the kidneys. However, the intracellular domain of nephrin is also really important for its physiological function because the intracellular domain promotes actin polymerization. And we know that the actin polymerization that occurs downstream of nephrin is absolutely essential for maintaining the really highly polarized and specialized cell shape that kidney podocytes require. So loss of actin polymerization is actually really detrimental for blood filtration and will actually lead to kidney failure in both human disease and mouse models. So how does nephrin regulate actin polymerization? It regulates actin polymerization by assembling a supramolecular cluster that contains multiple proteins. So there's really 3 proteins that are necessary for nephrin to promote actin polymerization. There's nephrin and the intracellular domain of nephrin in particular, NCK, which is a cytosolic adaptor protein that binds to nephrin, and NWASP is an actin regulatory protein and a nucleation promoting factor for the ARP23 complex. And what many labs have found is that nephrin co-localizes with NCK and NWASP in these micron-sized puncta on the plasma membrane of cells, and each puncta is capable of locally polymerizing actin filaments. So you can see in this image in a tissue culture model that expression of nephrin will cause the formation of these puncta that locally polymerize actin filaments. And so the Rosen lab was really interested in understanding using reductionist approaches to understand how nephrin actually assembles clusters and how these clusters polymerize actin. And so a former graduate student from the Rosen lab, Sudeep Bandrati, developed an in vitro reconstitution of nephrin clusters to try to really dissect the mechanism by which clusters form and polymerize actin. And so we can mimic nephrin clusters in vitro using supported lipid bilayers. And this is an approach that I use quite frequently and continue to use to try to reconstitute various signaling pathways. So we make supported lipid bilayers to mimic the plasma membrane. These bilayers contain trace amounts of a nickel chelating lipid, which allows us to attach his-tagged proteins directly to the bilayer. So these his-tagged proteins are permanently attached to the bilayer and able to freely diffuse within the bilayer. We can also fluorescently label our molecules of interest to visualize them using fluorescence microscopy. And so what Sudeep found was that in the absence of other proteins, nephrin is sort of uniformly distributed on the bilayer. But if he adds a sufficient concentration of NCK and NWASP to solution, he observes the rapid formation of micron-sized clusters that co-localize nephrin, NCK, and NWASP. And Sudeep found that these clusters form through liquid-liquid phase separation. So it was in fact the multivalent nature of these proteins that was necessary to enable cluster formation. Nephrin contains 3 tyrosines that when phosphorylated bind the SH2 domain of NCK. NCK contains an additional 3 SH3 domains, which each bind to one of the many proline-rich motifs in NWASP. So these multivalent interactions are sufficient to drive phase separation. And this phase separation promotes the formation of these clusters. So we showed that cluster formation requires multivalent domains. Having proteins with a single domain does not enable cluster formation. These clusters exhibit liquid-like material properties, so proteins are able to really rapidly and freely exchange between the cluster and the bulk solution. And the clusters also can undergo fusion, much like droplets would fuse, as you can see in this movie. And he also found that cluster formation was reversible. So if you add, for example, a phosphatase to dephosphorylate nephrin, the clusters will rapidly dissolve. So we have here a really simplified in vitro reconstitution of a signaling cluster that forms through phase separation and also has a defined biochemical activity. At least in cells, these clusters polymerize actin filaments. And so I was really interested in using this system to understand or ask whether phase separation was important for downstream signaling. So are there any molecular activities that arise from phase separation? And if so, how is this activity regulated or controlled? So the first thing that we did was try to develop a microscopy-based assay to visualize actin polymerization in these clusters. And so INWAS promotes actin polymerization through the ARP23 complex. So for this experiment, we add ARP23 complex and fluorescent labeled actin to preformed clusters. And so in this time lapse, you'll see that INWASP is in cyan and actin is in magenta. And what we found is that these clusters are sufficient for actin polymerization. When we add ARP23 and actin, we see really robust actin polymerization emanating from within the clusters. And so when we looked more closely at these images, we're actually really intrigued by what we saw, because we actually only ever see actin filaments initiating within the clusters themselves. We really never see any actin filaments polymerizing in these unclustered regions on the membrane. Even though there are INWAS molecules localized in these unclustered regions, they're just at a much lower density. And so this preliminarily suggested that there was something different about clusters that was enabling actin to locally polymerize within these micron-sized space-separated clusters. And so to probe this further, we're actually able to take these movies and quantify them. So microscopy is not only beautiful, but it's also highly quantitative. So we can measure the fluorescence intensity in these images. In different parts of these images, we can measure the intensity of each component, nephrin, NCK, and INWASP. We can also quantify the rate of actin polymerization to try to understand mechanistically what is happening within these clusters that is allowing actin to polymerize. So does enhanced INWASP activity arise from phase separation? And if so, how is this activity regulated? So when we did the quantification, we can plot actin intensity over time. And we saw that inside of these phase-separated clusters, we see really rapid actin polymerization, whereas outside of the clusters, there's very little actin polymerization. However, we also knew from our images that INWASP was a lot more concentrated within the clusters. So our first thought was that perhaps you simply see more actin polymerization because there is more INWASP. So to determine that, we actually were able to divide the rate of actin assembly by the local INWASP concentration. And this gave us what we call our INWASP normalized actin assembly rate. And we were actually really surprised to see that the INWASP normalized actin assembly rate was faster inside of clusters compared with outside of clusters. So it's not simply that there's more INWASP molecules in clusters, but actually each INWASP molecule in clusters is somehow more efficient or more capable of polymerizing actin than the molecules outside of clusters. And so this suggested that there was something different about the environment within a cluster that was altering how INWASP was able to promote actin polymerization. And so through a series of experiments, we determined that INWASP activity was not regulated by its density, but rather it was regulated by its membrane dwell time. So both NCK and INWASP are cytosolic proteins that transiently interact with the membrane through protein interactions. So nephrin is a transmembrane protein in cells, and in our in vitro reconstitution, it is permanently attached to the membrane. But both NCK and INWASP can bind and unbind the membrane through interactions with nephrin and NCK. So INWASP is sort of transiently recruited to the membrane and then dissociates and then rebinds and dissociates. And what we found was that inside clusters, the membrane dwell time, so the amount of time that an individual INWASP molecule stays associated with the membrane before dissociating, is increased. So it's about doubled the dwell time of the same INWASP molecules outside clusters. And this ended up being important for the regulation of its activity, because INWASP-dependent actin assembly requires a multi-step activation. So for INWASP molecules to promote actin polymerization, there are multiple binding events and multiple conformational changes that have to occur before an actin filament can be nucleated. And so what we think is happening is that the longer an INWASP molecule stays on the membrane, the higher the probability of successfully completing all of these steps and successfully nucleating an actin filament. So the longer INWASP stays on the membrane, the higher the probability of nucleating an actin filament, and therefore longer membrane dwell times increases the rate of actin polymerization. And so we were actually able to test this to generate clusters. We did many different experiments, and we consistently saw that things that we did to increase in-wasp membrane dwell time consistently increase its activity. But one experiment we were able to do, which is shown here, is actually change the membrane dwell time of in-wasp in clusters. So we could form clusters that actually have different stoichiometries of the components, so different amounts of nephrin, NCK, and in-wasp. And we were able to, through this, form clusters that have in-wasp either associating in the cluster longer or shorter. And we can measure both the in-wasp membrane dwell time using a photoactivatable in-wasp and the rate of actin assembly. And we see this really strong positive correlation between dwell time and the rate of assembly. So the longer in-wasp is staying associated with these clusters on the membrane, the faster the actin is polymerizing. And so we were really excited by this because we think through this in vitro reconstitution, we were really able to dissect what was happening in these clusters. But we also knew that this reconstitution was still it's a very simple system that's not quite as complicated as what's happening in cells. So we wanted to take these observations, this new hypothesis that we sort of identified that dwell time and activity correlate with each other, and see if we could observe the same behavior in cells with like the more complicated cellular environment. So we used a cellular system that other labs had established to study nephrin clusters in tissue culture. We can have the nephrin intracellular domain expressed fused to a CD7 transmembrane domain and a CD16 extracellular domain. And this system allows you to induce clustering using antibodies to CD16. So you can sort of artificially induce nephrin clustering. We have NCK and in-wasp fluorescently tagged and expressed in the cytoplasm. We can form clusters to measure the rate of actin polymerization. We did sort of a pulse chase type experiment where we can inhibit actin with latrunculin to depolymerize all of the actin filaments and then wash out the latrunculin for 5 minutes and fix the cells. So what we're measuring in our images is the amount of actin that each cluster was able to polymerize in a 5-minute period. And when we look in cells, we can therefore, because we have all of our molecules fluorescently labeled, we can see that nephrin, in-wasp, and NCK are colocalized within these clusters on the plasma membrane. These clusters are polymerizing actin, which we can visualize with phalloidin. And when we quantify the amount of actin polymerized in clusters, normalized to the in-wasp intensity, and in a separate analogous experiment, measured the dwell time of in-wasp in clusters of the same composition, we see, just like in vitro, we see in our cells that the dwell time of in-wasp, the membrane dwell time of in-wasp, the longer in-wasp is staying associated with clusters, the more actin we see polymerizing. So we think that this is a general way to control activity in phase-separated compartments, and this really interesting phenomena that we observed in our reconstituted system is also at play in cells. So we think that this is sort of a nice example of how the observations made in these simplified reconstitutions is actually can be really powerful for telling us what's happening in a cellular environment. So to sort of summarize what I showed you and some of what I didn't have time to show you, this is sort of our working model of how we think actin polymerization is being regulated within these nephrin clusters. In the absence of phase separation, signaling is really efficiently repressed because in-wasp is never able to associate with the membrane long enough to recruit an ARP23 molecule, induce conformational changes, and nucleate an actin filament. Upon phase separation, signaling turns on because you're now within the cluster able to keep in-wasp on the membrane long enough to nucleate actin filaments. And within the phase separated regime, we actually find that you can still tune the activity. We see this in vitro and in cells that there's a pretty dynamic range of activity within phase separated clusters because the relative stoichiometry of nephrin, NCK, and in-wasp ultimately can vary quite a lot. And it's that stoichiometry that dictates the in-wasp membrane dwell time and therefore the rate of actin assembly. So we think that phase separation is, I can't actually read what this says, actually can enhance biochemical activity. We also found that stoichiometry, changing the relative stoichiometry of components in a cluster is sufficient to tune this activity. And there's really only two features required for this mechanism, we think, which are multivalent interactions and a multi-step activation. And so those two features in a signaling pathway should be sufficient to see this type of behavior. And in fact, Jay Groves' lab at UC Berkeley saw really similar phenomena with RAS signaling downstream of T-cell receptor and LAT clustering. So we think that this could potentially be a pretty general mechanism that lots of signaling pathways could use to regulate activity. So that concludes sort of the first part of the talk, where really this was the first time we've really been able to show like a mechanistic functional consequence of having this type of phase separated assembly on the plasma membrane. But I was also really interested in exploring other signaling pathways that might, or other receptors that might also be regulated through phase separation, perhaps through fundamentally different mechanisms. So a lot of our lab and other labs had found a lot of these receptors that have multivalent tyrosine, sort of SH2 domain binding motifs on the intracellular domain that recruit these adaptor proteins from the cytoplasm to induce phase separation. So there's quite a few examples of this, with like the LAT receptor behaves similarly to nephrin. But there are lots of other different types of receptors that have very different transmembrane domains. And I was curious if any of them might also be organized through phase separation. So I also have been studying integrin receptors, which are a different class of cell adhesion receptors in mammalian cells. And integrins are receptors for the extracellular matrix. So they bind to ligands like fibronectin and collagen in the extracellular space. They are essential for multicellular life because they enable sort of tissue microenvironments to exist. So integrins bind to the ECM, and they also, similar to nephrin, sort of indirectly connect to the actin cytoskeleton and recruit a host of cytosolic adaptor proteins and signaling molecules to regulate downstream signaling. So integrins regulate signaling molecules such as kinases and Rho GTPases. They physically connect the actin cytoskeleton to the extracellular matrix, and they are sites of mechanical force transduction. So forces generated in the actin cytoskeleton are transmitted across this macromolecular complex. But unlike many receptors, integrins don't have intrinsic catalytic activity, and they don't bind directly to actin. So they're really entirely dependent on the ability to assemble a supramolecular complex in order to regulate their downstream signaling. And we know that integrins cluster with hundreds of different adaptor proteins to form complexes that are often called focal adhesions. And so these are micron-sized clusters. So this is a turf image of a protruding edge of a mammalian cell, and integrin complexes can be seen throughout the leading edge of this cell. So we know that focal adhesions rapidly form at the leading edge of cells. We know that these receptors colocalize and concentrate hundreds of different adaptor proteins that are cytosolic adaptor proteins, but we still don't really understand how these complexes initially form. They pretty much form in under a second, and there are at least 10 different proteins that spontaneously and instantaneously appear within these micron-sized clusters when they form. So I was really intrigued to explore whether phase separation could play a role, because a lot of the features of these complexes reminded me of phase separated compartments. They are inherently discrete micron-sized complexes. They have many, many multivalent adaptor proteins that are localized within them. They exhibit a wide range of stoichiometries, and they exhibit liquid-like material properties. So we had the hypothesis that phase separation of potentially multivalent adaptor proteins could promote initial focal adhesion formation. And we took an in vitro biochemical approach to test this hypothesis. So first, we just asked whether there were any focal adhesion adaptor proteins that were capable of undergoing phase separation in vitro. So I expressed and purified sort of 10 candidate focal adhesion proteins and screened them in vitro for phase separation. And what we found was we've so far identified 2 distinct sets of molecules that are each sufficient for phase separation in vitro. So when we combine, so NWASP and NCK also localize to integrin receptors where they can interact with the multivalent adaptor protein P130Cas. And these 3 proteins, when you combine them together, form liquid-like droplets that are phase separated. And we also found that the adaptor proteins focal adhesion kinase and paxillin were sufficient to form liquid droplets in vitro. So FAC alone can undergo phase separation. And when we add paxillin, this greatly enhances its phase separation. So to sort of briefly summarize a lot of in vitro work, what we've identified are 2 distinct branches, one that is FAC dependent and one that is Cas dependent, that each can induce phase separation in vitro. Furthermore, these molecules actually all interact with each other. So Cas can bind paxillin and FAC, but none of these proteins directly bind to integrin. So this alone we didn't think was necessary to cluster integrins. But what we found was that there was another adaptor protein, kendlin, that directly binds to integrin that could sort of connect these phase separating adaptor proteins to the integrin intracellular domain. And so what we find is that if we combine either branch of these pathways, the P130Cas set of molecules or the FAC set of molecules, we can form droplets that concentrate integrin intracellular domains. But only if we add all of these molecules together, we find that all of these proteins can colocalize, and we see a really strong enhancement of integrin partitioning into these droplets. And so we thought that with this set of proteins, this should be sufficient to reconstitute clustering on a supported lipid bilayer using a very similar assay to what we used with nephrin. So if we attach integrin to a bilayer, what we see is that in the absence of other adaptor proteins, we don't see any clustering of the integrin receptor. If we add either the FAC-dependent branch or the Cas-dependent branch, we start to see some clusters form. But when we combine all 7 proteins together, we see really strong clustering of integrins on a bilayer. And so this is quantified here, where there's this really strong cooperativity between P130Cas and focal adhesion kinase in promoting integrin clustering. And so this was really interesting to us, and we thought this is a prediction we could test in cells. If what we're observing in vitro is important for how cells regulate integrin clustering, we should be able to observe this cooperativity in focal adhesion formation in cells. And so to test this, we knocked out either FAC or P130Cas, or we knocked out both FAC and P130Cas together. And what we find is that in the absence of either Cas or FAC, cells form fewer focal adhesions, but they still form focal adhesions. But when we knock out both FAC and Cas, there's a really strong defect in focal adhesion formation. So cells really fail to spread because they can't form focal adhesions after 30 minutes period. And so this is all sort of suggesting that we think that there's this a lot of redundancy in the phase separation of these proteins, but they also cooperatively function together. So FAC and P130Cas can both promote phase separation to cluster integrins. They also cooperatively can interact with each other to enhance focal adhesion assembly, and that perhaps phase separation is one mechanism that can serve as an intracellular trigger to promote initial focal adhesion assembly. So as I start my lab at MIT, I'm really interested in continuing to pursue use this reconstitution of focal adhesions to better understand, again, if there's a functional consequence for this phase separation in the downstream signaling of these proteins. So is there any functional consequence for phase separation at focal adhesions, either for their chemical signaling, so the regulation of kinases and rho-GTPases, or the mechanical signaling, so the forced transduction that is occurring. So if focal adhesions can be thought of as a liquid phase that connects the actin cytoskeleton to integrin receptors, some of the questions that we're currently pursuing is trying to understand if phase separation regulates FAC kinase activity. So we've identified a kinase, focal adhesion kinase, that is sufficient to form liquid droplets in vitro. And now we're trying to understand how its autophosphorylation and the activation of its downstream signaling might be regulated through its phase separation. We're also really interested in understanding the material property of this condensed phase and how forces might be transduced through this condensed phase. And understanding how both actin and mechanical forces generated in the actin cytoskeleton might alter or regulate the condensed phase. So I've showed you today two examples of the nephrin receptor and the integrin receptor being clustered through phase separation, and also potentially, at least in the case of nephrin, its downstream biochemical activity, the ability to polymerize actin, is enhanced through phase separation. But I don't think that this is really as super, there are many different receptors that could potentially be regulated through phase separation. So phase separation could be a much more general mechanism to organize receptors on the plasma membrane. So we see that nephrin can be clustered through phase separation, integrins can be clustered through phase separation, but there are many other clusters and diverse cell types that us and other groups have observed can also be clustered through phase separation. So the LAT receptor in T cells, the Wnt receptor in disheveled, ZO1 and Claudins at tight junctions, receptor tyrosine kinases, and postsynaptic densities and NMDA receptors. There's growing evidence that this is a really common phenomenon at the plasma membrane. So I think understanding how signaling is regulated within these types of liquid-like compartments that form on the plasma membrane is a really important fundamental cell biology problem to really understand. And I'm also really excited to understand how phase separation driven by protein-protein interactions is sort of regulated in combination with other factors like cytoskeleton or lipid composition or carbohydrate modification, all of these other factors that we know also regulate molecular organization at the plasma membrane. How are all these things functioning together in cells to organize molecules at the plasma membrane and regulate downstream signaling? And so with that, I will conclude, and I would like to thank you for your time in another virtual seminar and your attention. Mike, for being a fantastic postdoc mentor, and all of my former colleagues in the Rosen Lab, Jay Groves, who I was really fortunate to work with for a summer in Woods Hole, where he helped me and Mike really understand their ideas about kinetic proofreading at the plasma membrane. And my lab, so my growing lab at MIT, this is our first group that we had over the summer who've been sort of diving into exploring phase separation and focal adhesion proteins. So thank you, and I would love to take questions.
Thank you, Lindsay. This was amazing and very well attended, by the way. We've done very well. We peaked at 65 attendees, and I know that this is kind of later in the day, but we have several questions, some of which arrived in various routes to me because we had a little mishap with the Q&A for a few minutes. But so questions coming your way that I will read out to you. So one of the questions that refers to the first part of your talk was asking if NWAS dwell time increases in the clusters, as you showed us, wouldn't that also increase their concentration? So how are you able to separate basically these parameters?
Yeah. So yeah, it would increase their concentration. So we're always normalizing the activity to the concentration. So what we're measuring is sort of the concentration, the activity per NWAS. So we did a series of experiments where we varied only the concentration or the density, and we saw like no correlation. There's basically, you can increase the density of NWAS on the membrane by orders of magnitude, and the rate of polymerization per NWAS is constant. So we thought it was density to begin with. Now that doesn't say, there is also potentially like a difference in like how the molecules are organized, or some, like, there are potentially other, like the pore size could be changing. So we do think there could be something potentially that's correlating with dwell time that perhaps we're not realizing, but we are pretty confident that density isn't really alone a key factor.
That makes sense. Another question is actually my question, was about the actual lipid composition of the bilayer that you're building. And I was first curious about its composition and how you choose it to be the way it is. And then also, would any of your outcomes or measurements be altered if you altered the lipid composition, and in what ways?
I'm very interested in that question. So currently, for technical limitations, technical reasons, it's a very simple bilayer. We just have phosphatidylcholine and a little bit of PEG, which helps the bilayer stay fluid and not like get adhered to the glass, and then nickel, which is nickel lipids, or how we attach the His proteins. So it's a very artificial bilayer in that it's like not at all what the plasma membrane is like. So really, in our system right now, the bilayer is just sort of a thing the proteins are attached with and diffusing on. So it's important that it's fluid. If it's not fluid, this doesn't work. But I know that, like, for example, with the focal adhesions, there's a ton of those proteins bind to PIP2 phospholipids. And so a major question I have is, like, can we introduce phospholipids, phosphoinositides into the bilayer and understand how lipid composition could be really important? That's really hard to do the way we currently do. We've tried, and charged lipids want to interact with the glass, which prevents the bilayer from being fluid. So we're working on developing different strategies for making bilayers with more complex lipid compositions. Also, we know when you have really complex lipid compositions, sometimes the lipids themselves spontaneously phase separate, which can be interesting, but also sometimes artificial to in vitro work. So yeah, I think that's just one of the most fascinating questions about this happening at the plasma membrane, is this potential for, like, sort of cooperativity between proteins and lipids, and how lipid composition plays into this is a really interesting question, but we don't know.
Yes, yeah, to be determined.
Yes.
More questions that have come in. Wanchi from my group actually wants to know, do the early kinetics of focal adhesion formation indicate that phase separation explains that entire process?
Yeah, I don't know. So I mean, no, it can't explain the entire. So the other half of this that I'm ignoring, both in the nephrin and the focal adhesions, is like the extracellular domains and what they're doing. So we know integrins bind to the ECM, and we know that they have to undergo a conformational change to become active to bind the ECM. And so what I don't know is how activation might be coupled to phase separation, or that relationship, and what the extracellular domain is doing, which we know is important in cells, of course. Yeah, I don't know if it's, and then with integrins, there's the long in the field, there's this idea of inside out signaling versus outside in signaling. So that's sort of like initiating clustering from the inside of the cell versus the outside of the cell. So I sort of think of this as potentially like an inside out way to initiate formation, but certainly we know that's not the only way to initiate the process. So yeah, I guess I don't know, but it would be interesting to think about that some more. Yeah.
I understand that Ed Skolnick, who's listening, had a question, which was regarding the first part of the story. Again, the complex that you described, is this complex in the glomerulus? He's asking.
I don't know off the top of my head. So yeah, nothing that we've done is like in an actual kidney, so we don't have like physiological structures forming in any of our experiments.
But I actually found your work very meaningful, because from our own work in the filter, actually all of the components that you have there are very important for the formation of the filter. So they are part of the glomerular structure, and in fact, there are several mutations in patients who have severe disruptions to their kidney filter. So I thought that work was beautiful, actually, and it really recapitulates. I
mean, I know there are many components missing from what we've reconstituted that are important, but I think, that's good to know.
Yeah. Another question is, what is the time scale of the clustering? Is this a relatively fast or slow signaling process?
Yeah, clustering is very, like within seconds you form clusters. So it's very fast and reversible. So that's one of the really, I think, exciting things about phase separation is it gives you really large changes in a really fast time scale. Yeah.
And another hard question that, of course, is informed by, my own interests in occasionally in proteins that are multipass. So is that really something feasible for down the road, for being able to really study those types of signaling complexes?
Oh, yeah, like that's technically harder of it. Yes. So I haven't even had like a single transmembrane domain in any of my in vitro assays. That's something I'm really interested in doing. I think it would be really interesting to try to
even a single transmembrane domain.
A single transmembrane domain, I think. And then it's like, how do you control the direction of insertion of these things? So I think with like, I've been looking a lot at, there's like these new technologies to sort of extract proteins from the membrane, maybe in mammalian cells, and then maybe you could like fuse that with the vesicles we're making in vitro to try to deposit them on the membrane. But I think that would be very challenging, but very cool, because like most things are. But yeah, I think starting with a single transmembrane domain would be challenging enough. But then if that can work, then we could potentially look at others.
Very good. We already answered all the other questions, I think. Ed was interested in the diseases, and I mentioned that there are diseases that correlate with that. So it only makes the importance of this work greater, in many ways. Actually, there are, as far as the kidney goes in particular, there are mutations both in integrins and in nephrin that lead to kidney disease. So this is highly relevant to certainly those structures. But I mean, integrins, of course, are relevant to every other cell biological process I can think of. And then if you start to think about neuronal spines and NMDA receptor signaling and other things like that, there's so many infinite really possibilities from the generalizability of this approach, really this scientific approach that you're proposing. So that's really great. I think this brings us to the end of our question and answer period. I want to thank you very much again. Oh, I'm very sorry, one more question. Do we really comprehensively understand, Jerry Yao wants to know, do we really comprehensively understand the protein components of those droplets in vivo? What is the current systematic method, if there is, to characterize their composition?
Yeah, so I'm not sure about nephrin, but with integrins, we actually have a lot of information from cell culture, I guess. So more in vivo than what I do. So people have there's been quite a lot of mass spec studies where people can purify focal adhesions and mass spec the components. And we know there are hundreds of proteins, and we also have seen their composition changes really dramatically under different conditions. So people have done studies looking, at different stages of the cell cycle, or obviously, on different ECMs, they're going to be different with a high rigidity substrate versus a low rigidity substrate. So the composition is really dynamic and very complex. And so there is a lot of information. And so that's one reason I think actually this is a great system to have a reconstitution, because now we can actually try to make sense of all like these lists and lists of proteins, like what are all these proteins doing? But yeah, so the proteins that I sort of screened, it was a very biased screen. I picked proteins that were necessary, like we knew were necessary for function. But I think almost a more interesting question is like, what about all the 100 proteins that we don't know anything about their function? So I'm really interested in taking advantage of all of the proteomics work that's been done on these complexes. Yeah.
That makes
sense. And we also know from evolution, there's 12 proteins that seem to be like really conserved with integrins that are, through like C. elegans and Drosophila, but then there's in humans like 100 more that, and lots of gene duplication events as well. Yeah.
Tremendous enthusiasm. So one more question actually that just came in. She wants to know, you've mentioned that phase separation is reversible. Does this mean the condensates get smaller or fall apart completely? And in either case, what does this mean in the cell?
Yeah, so they in vitro, they fall apart completely very quickly in vitro. We can do things in cells that's very artificial. So there's this alcohol 1,6-hexane diol that can often really rapidly dissolve phase separated structures. And when you add that to cells, obviously there's a lot of things that get messed up. So it's not the most elegant experiment, but for example, I can add that to cells and focal adhesions immediately dissolve. However, we know the turnover of a lot of these complexes is actually tightly regulated. So it's not like they're just dissolving. There's actually proteases and events that sort of disassemble these structures. I think mitosis is really interesting. A lot of different components get dissolved during or go away during mitosis, both this like cytoplasmic puncta, the nuclear puncta, focal adhesions, like all of these things have to like rapidly go away to divide the cell. And so that's potentially an interesting place to like maybe, you know, under certain points when you need a really quick global dissolving of something, that could be a way to do that. But yeah, so I think there's more, I think a lot of work has been done on formation, less on the dissolution. That's just as interesting and important. Yeah.
Of course. Sounds terrific. Well, this brings us to the top of the hour. So thank you again so very much for being with us today.
Thanks for all the questions and for everyone's attention.
Absolutely. Thanks again. And thank you to all of our listeners for participating in today's seminar series. So thanks again very much, Lindsay, and thank you all. Have a good afternoon.